And then at the end of the session, we'll have about 15 minutes for the audience to ask their questions. So we are going to start with Erjola B. Hold on a second. Yep. Okay. 1 second. Okay, so yeah, first of all, we have Erjola Pe. She's a PhD student in clinical medicine and public health at the Department of Life, Health and Environmental Sciences at the University of L'Aquila in Italy. And she is also a member of the Department of Chemical-Toxicological and Pharmacological Evolution of Drugs at the Catholic University of Our Lady of Good Counsel in Albania.
Thank you, everyone. It's so nice being here. Not this one, the other one, please. Okay, so as it was said before, my name is Erjola Bey. I'm a PhD student. I study medical sciences in the University of L'Aquila. And yes, thank you. Okay, my research project is about saffron, and because we think that it might have many properties, such as anti-inflammatory, antioxidant, immunostimulant, anti-cancer, and antidepressant. First of all, we started our research project with a questionnaire because, since I come from Albania, we wanted to know how many Albanians know of saffron and its properties. The data of the questionnaire were completely confidential. 780 Albanians completed the questions, and the results are reported in the charts below. As you can see, most of those who completed the questionnaire were female, 80%. And then the other part, the other question regarded the ages. Most of the people who completed the questionnaire are between 18 and 30 years old. But there are also older people from 30 to 45, 45 to 60, and older than 60 years old. Most of the people who completed the questionnaire are from Tirana, which is the capital of Albania, and the others from other small cities such as Durazzo, Scutari, Valona, and other cities in Albania. The education the people, all these 700, almost 20 people have, is in most of the cases, as we can see in the charts, is higher education. There are also some people with graduate studies, school, high school education, and basic education. The professions are medical professions, economists, educational professions, engineers, and most of the people who have completed in the meantime are students. We also wanted to have a question about the incomes. This is only because since saffron is considered to be the red gold, so it's very expensive, we wanted to know if the people in Albania having a low salary can afford it. So as we can see, there are people with low salary, low medium, medium, and medium high, and high also. The percentage of people who have heard of saffron before is 72%. All the other results that are going to be presented in the other graphics are the answers given by these 72% of people, which were almost 520 in all who know of saffron. So we did not take in consideration the other percentage, almost 20%, who had not heard of saffron before. So most of the people think that saffron is used in cooking, and it stimulates the digestion, and it also has a cytotoxic effect. In the meantime, there are people who do not know the daily dose of saffron, both in adults and in children, as we see in the first graphic. And since some of the people who completed the questionnaire are people who were of the medical professions, they knew that saffron can interact with other medicines, such as, for example, anticoagulants and other drugs as well. Saffron might also have some side effects if we get more than the daily dose or if we get more than what is prescribed by the doctor. The second step of the research project was taking a sample of saffron from this Italian-Albanian company which operates in Albania since 2013. And in the pictures we can see all the process of the cultivation of the saffron, starting from the bulb and going on with the essiccation process in the dark, and then gaining the final result of the saffron. Saffron itself can be classified in 4 categories based on the absorption. The Albanian saffron, based on the results of the analysis that we got from Italy, is classified as the first category, which is considered to be the first, the best category. Then the third step of the research project regards taking this sample of saffron and preparing with it a hydroalcoholic solution. This solution is put in the dark, and it's agitated for almost an hour. After that, we put it in the centrifuge for 10 minutes, and we get these 3 small tubes, which are, as we can see in the other slide, which are then, with this surfactant-free cellulose acetate filter, as we can see in the picture, are filtrated and divided into 4 small tubes. These 4 small tubes, before having the concentration, are empty weighted before, so we see the mass of the tube being empty. Then all these 4 tubes go during all the night in the fridge. The next day we put these 4 tubes in the speed vac. Speed vac is a machine which is normally used to concentrate small samples and to eliminate the liquid. So in the end we just get the precipitate part. We put the 4 tubes with the tape open, so ethanol that we used at the beginning can be eliminated. We put it in most cases for 2 hours. If after 2 hours we see that there is still some liquid left, we repeat the process all over again. In the end, the samples must be completely dried. Then we weight the tubes again, making the differences before the tube with the sample and the first tube which was empty weighted. What we got is the precipitate of the polyphenol. Polyphenol is antioxidant, and we are using it in the next steps for in vitro model of neuronal senescence. This is going to be the next step. In the meantime, the polyphenol is put in fridge in minus 20 degrees, so it can last for a long time. As I said, I'm the PhD student of the University of Aquila, and the other 2 professors are my co-helpers in this research project. And I would like to add one last thing. This project research is contributed to the founding from the Catholic University, Our Lady of Good Counsel in Albania. That's all for the moment. Thank you very much.
Thank you so much, Ariola. Up next, we have Arnau. Arnau Vila is a bachelor student in biomedical sciences at the University of Barcelona in Spain.
We are running out of water. For the past few years, Catalonia's clean water reservoirs have been emptying at an alarming pace and have become incessantly polluted by intense anthropogenic activities. A relentless drought has forced the regional government to implement a state of exceptionality, and it has left the remaining waters heavily polluted by nitrates, affecting 3 million people, almost half of our population. But we found a way to change this.
This year, as the University of Barcelona's item team, we are developing algagenics. Our main goal is to eliminate nitrates from water using algae, whilst producing a high-value biological compound, cytokinins.
Cytokinins are of special interest because they are phytohormones, natural stimulants that induce cell division and thus enhance plant growth.
We're modifying Chlamydomonas cerevisiae, a microalgae, to increase their ability to assimilate nitrates by enhancing the expression of several endogenous genes.
Then, we are linking the absorbed nitrogen with the also enhanced metabolic pathway of cytokinin production by adding an exogenous and unregulated key enzyme.
Our algae is designed to fit in a bioreactor right within highly polluted nitrates water, from where our engineered Chlamydomonas will transform said nitrates into organic matter enriched with cytokinins. Then the algae are lysed and killed, and the final liquid product can be applied onto fields as a natural fertilizer. Algagenics has the potential to revolutionize a relatively unexplored field and to help solve a problem that it's not only affecting a specific region, but it's actually affecting millions of people worldwide.
We can revert the water crisis.
We can make a difference.
We are algagenics.
No, the second one. So, as the video told, we are algagenics. We are developing a project to present in a competition called iGEM. That is a competition that is celebrated worldwide, and it has the giant jamboree that is in Paris, in France. And we are the team that we are representing our university. This is our team. We are all a group of students from Biomedical and Biotechnology. And we all wanted to search for a problem and solve it using synthetic biology, because iGEM is a competition about synthetic biology. So we thought that there was a big problem with our waters, because we are, as the video said, we are running out of water, and the remaining water is polluted by nitrates and among other contaminants. But the main ones in Catalonia are nitrates, and we saw a lot of news stating that. And so we did a study using different databases, and we found that almost half of our population is affected by this lack of free of nitrates water. But where does the nitrates come from? They all mainly come from farms, agriculture, and industry. These are the main, the main pollutants. So, and they all end up to our water reservoirs that we use them to have clean water and be able to drink them. And why are these nitrates so bad? They are bad because if we ingest them, they can oxidize the iron molecules from our blood, and this will cause that the oxygen transport through our bodies will be slowed down. And it has been studied that an average person of 60 kilograms can ingest like a maximum of 219 milligrams of nitrates per day. That's a very small amount compared with the amounts of nitrates that there are actually in lots of water reservoirs in Catalonia. So it's an urge to clean all these nitrates. So for that, we designed a project. And after searching the problem, we thought that we could modify microalgae, because microalgae have the ability to absorb nitrates. So maybe enhancing these metabolic pathways, we can like absorb more nitrates and contain them in a photobioreactor because of the laws about transgenic organisms that there are nowadays. We can't just leave the algae alone. We have to have them controlled inside a photobioreactor. But we knew that there were some solutions about that, so we wanted to do something better. And what we thought was to make like a recycling project, so we could like absorb the nitrates and convert them into something that is for an interest. And after searching a lot of bibliography, we found that cytokinins that are phytohormones could be of a great interest because they are nowadays expensive to produce. So if our algae could produce them, and using all these pollutants, we can have more fertilizants of high value. And in this way, it's not just absorbing the nitrates and then another one would add these fertilizers to their fields, but it's like recycling the same nitrates and not creating more fertilizers. What? So to do that, first of all, we did a prototype. We looked for the genes that we wanted to enhance. And about the nitrate assimilation pathway, we found like the two major genes that are NR and NIR. And also we had to regulate the GS. And for the cytokinin production, there were also two genes involved in that were IPT and LOG. So after finding them, we just did the synthetic biology part, that was to do the assemblies. And for that, we used MOCLO, that it's a technique to use synthetic organisms, especially used in Chlamydomonas. So we designed like the level 0, level 1s, and level M's. That is like different steps of the genetic modifications. So first of all, the most important parts are the level 1 and level M, because level 1 is like the gene, but with high promoters, so the gene will be overexpressed and the algae will be able to express more and more of this gene. And the level M is just to insert them into a plasmid with a resistance, and also the M-cherry and M-venus that are reporters so this level M is the one that is inserted into our Chlamydomonas, like we can see, and the Chlamydomonas are plated, they grow, and then when there are the colonies, we can just put them into Erlenmeyers and grow them at a big scale. And then we will be used to do the further experiments that we haven't reached yet, but because we are now in the lab studying it, so maybe you can have some results in over a month or like this. And at the end, we thought, okay, what will be the production of our algae? And we found that if we had 25 liters of polluted water with nitrates and using only 10 kilograms of microalgae inside a photobioreactor, we could have the water clean and enough cytokinins to fertilize 1 hectare of fields. So that could be a great number. And this is algagenics. But for that, there's all the scientific part, but we had to approach our project to society. And we found that the people we had to talk with were like farmers, agricultures, industries, and a new field that are hydroponics, that are like, they do plants and food without field. And we have been talking with them, doing like interviews in order to approach our project, and also speaking with different specialists about microalgae and all this world. And they all got to the point that we had to bring our project also to space. And we were like, why? They all said that, okay, in Earth there are like these regulations, that maybe using a transgenic algae, nowadays it's like impossible, but in space there are no regulations. And it's also a high, they also need this because they all need, for example, in the space station, or even if humanity would colonize Mars, they will need to clean waters and have also fertilizers to produce their food. So they said, why don't you explore also the space? And it's just a thing that we mentioned because it was like a funny thing. But mainly, what we've been doing now is like talking with the experts from farming, agriculture, and hydroponics, and also experts about microalgae. So we are focusing our project right now with these 3 big fields. And that's all for my part. Thank you very much.
Thank you so much, Arna. Up next, we have Milena Stepic. She is a student at the Faculty of Biology in the University of Belgrade, Serbia.
Just turn on here. Yep. It works a bit, yeah? So you can hear me? Okay. Hello, everyone. Thanks for being here. My name is Milena Stepic, and I've just finished my 2nd year of studying molecular biology and physiology at Faculty of Biology in Belgrade, in Serbia. And today, I will be presenting my project that I've conducted under supervision of Dalibor Štis, a scientist employed at the Institute of Complex Systems in Czech Republic. Given that in molecular biology, that is my field, the principal thing when we want to study something, to investigate, is cell along with its genetic material. There is an obvious need for a development of new method for cell visualization, new kind of microscopic techniques that are fast, simple, and that can be done without previous fixation or staining or any kind of complication. So in my project, I've been using a specific microscope, Futurescope, constructed by the Institute of Complex Systems, where I've been doing all of my research. And I've been filming HeLa cells that you can see in the background a bit. What was the main goal? I've been using this Futurescope to film HeLa cells and then obtain a cell spectra from those cells that I've been filming. I will describe later a bit about cell spectra. And then when I obtain cell spectra from HeLa cells, the main goal was to link those spectra to the exact phase of the cell cycle. So I need you to first understand, this was the first image I've obtained using Futurescope. So here you can see like a bunch of cells. And what I've been doing, I've been trying to separate them into 3 groups according to their physical appearance. And the first group here represented cells that had that roundish shape. And I assumed that they are about to divide. So they're at the start of their division process. The second group here, those cells are almost finished with the division process. You can see that they're almost divided. So this was the end of the division group. And this one represented the cells that are not currently in the division process. So that is interphase. So once that I obtained those cropped out image, I cropped out this little picture from this huge one. I've been using specific algorithms that were connected directly to the futuroscope to extract some informations. At first place, I've been using SpecV2, which is that super cool software that can extract from each pixel that are present in one image. It can extract those cell spectra. So it would be like transparency in the function of wavelength, and so each pixel has its own spectrum here, and later I will be naming it spectral parameter, but I will discuss about a bit later. So it was SpecV2 at first place, and at the second place it was a specific algorithm written in MATLAB. And firstly, I need to introduce you to the concept of clusters. So clusters are groups of pixels with similar spectral properties. So firstly, I've been doing SpecV2, then I extracted cell spectra, and for each of pixel, for all of those pictures, just a triplicate. And then spectra that are similar, that has pixels that have similar spectra, they are grouped together and given here in the same color. So, for example, if you look at this area that is orange, it represented one group of pixels that have similar spectra. So they are called clusters here. And also, I've been calculating what is happening with the localization of those clusters within all of these three groups. That is, I've been trying to estimate is there any kind of change location, and yes, that's all for clustering. And now I've been talking about what did we get at the end. So here, firstly, I need you to describe those graphs. As you can see, at first place I have clusters 2 and 4, and clusters 5, 9, and 7 and 9. Clusters 2 and 4 represented this kind of light and dark blue structure that was appearing along all of these three groups, but it was changing a lot, its size and shape, and I was interested to see what is happening when we compare each group with each group and somehow present it at graphs. And the second group, clusters 5, 7, and 9, they represented cell borders, which is cell membrane here. So you see spatial parameter and spectral parameter. So spatial parameter relates to the localization of clusters here, and spectral parameters refers to the spectra. So here, for clusters 2 and 4, we show that there is much more differences when we talk about spatial parameter. So you can see all of those colors. I will try to explain it. This group here, that will be start of division, is red colored. This one, end of division, is yellow. And this one here, interface, is blue colored. So here in graphs, for example, purple, then it means that I've compared red and blue group, and so on for everything here. And I've been comparing all of those groups, and we show that in spatial, when we talk about localization of those clusters, we have much more differences. When we talk about its chemical composition, that is cell spectra, so it means that it changes a shape, but not that much composition in cell. And when we talk about cell membrane, that are clusters 5, 7, and 9, we show that there is pretty much differences both in spatial properties than in spectral properties also, which means it changes its spectra at first place, but also its localization, its shape, which is logical because we were talking about cell division at first place. So it is logical that cell membrane changes a lot its shape during that kind of process. So what can we conclude at the end? We can of course, conclude that different phases in cell cycle have different spectral and spatial properties, which I've been describing here, okay? So it is especially noticeable in the group of clusters 5, 7, and 9 that was representing cell membrane. And it is, of course, still work here. One of my idea was also to add some substances. For example, in my example, it would be concanavalin A. It's a kind of substance that will push my cells to divide more. So I've been interested in to see what will happen when we compare groups after some period of time with control. What will happen with those spectra? Will it change or not? And what will happen with groups of pixels? What will be at the end? But it was my extra idea, which I will be maybe in future, investigate maybe in future more. And also, what was the main point? At some time, if we investigate enough, we will be able to have a picture of cell, and using FutureScope, using those algorithms, maybe say, okay, this cell has spectrum that will tell us that cell is about to die, the cell is about to divide, to tell anything about its physiological state, to say it like that. And then once that we can do that, I can say that we will have a fast characterization rapid method that can easily bring us any information from just using bright field transmitted light microscopy, which was my idea at the very beginning of this research. So thank you for your attention once again.
All right, thank you so much. Up next, we have Antea Kršek. She has a bachelor's degree in physiotherapy and currently a student of medicine at the University of Rijeka in Croatia.
Thank you. Hello, everyone. My name is Antea. As colleague introduced me, I'm a medical student at this point, and this research was conducted in collaboration with my mentor, Professor Lara Baticic, who is professor at the Department of Chemistry, Biochemistry, and Clinical Chemistry at University of Rijeka. So the topic today is basically the silencing of peptidase 4 in experimental colitis, which will disrupt the concentration of vasoactive intestinal peptide within the gut-brain axis. So we are referring more to medical pathophysiology. So I'm just curious how many of you here is like medical students? No? Okay, then I will try to keep it very simple and just possibly interesting for you. So first of all, I would just... Okay, so this is a short overview of slides that we will go through about this research. And just for clarification at the beginning, inflammatory bowel disease will be chronic and inflammatory idiopathic condition which currently doesn't have a cure, and the mechanism behind the disease is still quite unclear, but we are still working on it, and it includes ulcerative colitis and Crohn's diseases, which are both very disruptive in normal life and quality of it. So background of study was actually directed toward the just explaining how maybe this peptidase 4 will actually influence this inflammatory processes in the diseases. So we started with the setup, and we used two types of mice. So we had a wild strain and just the CD26 mice that was like resistant to it, and then we basically induced Crohn's disease into the mice through our experiment, and we studied how they will react and when the inflammatory process will start. We noticed the first changes within the 24 hours, and then we sacrificed a group of mice, 10 or 8 of them, every 2nd, 7th, 10, 15, and 30 days since the induction. And the results, we analyzed our results through vasoactive intestinal peptide. We used Western blot and ELISA tests to actually see what will happen if there will be any influence on both strains or just one. What we actually found is that the mice that were without CD26, they had higher levels of vasoactive intestinal peptide in their system, and they had different stages of inflammatory processes that were also presented on their brain. And this was the main results of this research, that we actually proved that the vasoactive intestinal peptide and induced Crohn's diseases would actually have effect on the brain and on the gut-brain axis. These are some pathophysiological results that we saw, and the first one, the picture A, this one, is actually how the normal tissue should look like. And then basically from B to D, you can actually see how the colons will have symptoms like acute inflammation, histology findings. Also, you can have, we saw mucosal ulcerations and neutrophilic inflammations in lamina propria. And then on the last pictures from E to G, from E to G, you can also see the colon sections after 7 days, since we induced CD26, and you can see like the inflammation, and submucosa can also be seen with necrotic parts that also will have fibrotic tissue. These are also some of the results. So the first graph here will be of the serum that was taken from our mice. And you can see the CD26 resistant type and the wild type. And these are the representations of vasoactive intestinal peptide concentration at different time schedule during colitis development. So this was serum. You can also see here the same for the brain and for the colon. This would be the brain and this would be the colon. And to conclude, I already explained why this was important. The discovery is basically because we discovered that the neuropeptides and interleukins will actually be changed in colitis, and particularly in CD26 deficient mice. We will highlight the role that it plays in brain and gut axis in IBD. I really hope that this was meaningful to you. I know that you're not medical students, but I think like you are biotechnologies and stuff like that similar. So you're doing these things, and I really think that stuff like this, and you're an important part of researches like this that will actually play important role in our clinical part that we are actually doing as medical professionals. Thank you, and
All right, thank you so much, Antea. And now we have 15 minutes in case there's any questions from the audience for any of the speakers. All right. Okay, I don't think there's any questions, so we can conclude this session over here. Thank you so much for participating, everyone. And thank you so much for the speakers for presenting their research with us as well.